Today we're going to explore onion cells versus cheek cells under the microscope. This is the basic structure of plant and animal cells. Both of them have nucleus, cytoplasm, and cell membrane. Plant cells have additional structures like cell wall, chloroplast, and vacuole. So let's view animal cells and plant cells under a microscope. For the first experiment, we will use the epidermis of an onion to view the plant cell. Here are the things that we will need: onion, iodine solution, distilled water, slides and cover slips, white tile, forceps, mounted needle, knife, and microscope. Cut the onion into 1 centimeter thick slices. And from one of the onion squares, we will take the epidermis. Put a drop of water in a slide and carefully place the epidermis. Cover the onion with a cover slip. Be careful so that there will be no air bubbles trapped. It might reflect the light and can obscure the image. Use a filter paper to absorb the excess water. Put a drop of iodine solution to stain the cells so that we can see the different structure of cells. And you can use a filter paper to absorb the excess iodine. You can also put the iodine in the epidermis first before covering it. Wait for 1 minute. Place the slide in the microscope. Select the objective with a powerful lens and focus. So what can we see? So we see the group of cells. The solid outline is the cell wall, and we can see the nucleus of the cell. And there are black circles are the starch granules which has stained black because of the iodine, and some of the air bubbles that is trapped. Now let's increase the magnification using another objective. Here we can clearly see the nucleus of the cell, the cytoplasm, and the cell wall. We cannot see the chloroplast because this part of the onion where this is planted underground, there is no photosynthesis. Now let's go to animal cell. We will need the following: cotton swab, methylene blue, empty microscope slide, cover slip, filter paper, microscope. We will use the cheek cell for this experiment, so gently rub the cotton buds inside the cheeks. Smear the cotton buds in the center of the blank slide to transfer the cell. Then, put a drop of methylene blue where the cheek cell was transferred. Gently put the cover slip in the slide, avoiding any trapped bubbles. And then dry the excess methylene blue using a filter paper. Then you will wait for 1 minute. So set up and focus the microscope like before, starting with the lowest powered objective. As you can see, animal cells are often smaller than plant cells. So it means we have to use a higher magnification objective lens so that we can see the cells in more details. So when we use the higher magnification objective lens, we can see the nucleus in the middle of the cell. And we can also see the cytoplasm. So let's now compare the two cells. This is the chick cell and the onion cell. So based on the outcome of the experiment, onion cells are joined together while the chick cells are separated. The onion cells are larger than the chick cells. Onion cells have a roughly rectangular shape while chick cells have an irregular shape. And onion cells have cell wall, but chick cells don't have. Thank you for watching. Don't forget to like, subscribe, hit the notification bell, comment, and share.